We've been talking about bacteria, but in this video today we're going to talk about bacterial reproduction and exchange of genetic material within the bacteria. Now, there are four different processes we're going to discuss: binary fission, bacterial conjugation, bacterial transformation, and bacterial transduction. And we'll start with binary fission. Binary fission means two parts, binary, to split into two. So here's our bacteria cell. We see that it has this one main circular DNA or chromosome. And in the first step of binary fission, we have to replicate this chromosome. So we make an exact copy of the chromosome. Then the cell is going to start to elongate. And as it does, it pulls those two copies of the chromosome apart. And eventually and finally, it divides into two. Now, this is reproduction for bacteria, and it's asexual, meaning that we need no gametes. We don't have to produce any special reproductive cells, and we don't need a partner. One cell splits into two. And these two cells are genetically identical to each other. So while binary fission can create a very large number of bacteria in a very short amount of time, it doesn't create a lot of genetic diversity within our bacterial populations. Outside of the chance random mutations that happen during DNA replication, we're not going to create any other diversity. Now, under optimal conditions, the right temperatures and the right nutrient material, bacterial populations can rapidly divide. In a very short amount of time, you could have a very large population of bacteria. So we can summarize the process visually here. We can see kind of visually what's happening. And so binary fission is the asexual reproduction of bacteria, creating new bacteria, but very little, I shouldn't say no, but very little genetic diversity. So how do we get genetic diversity within bacterial populations? The answer will come in these other three processes. So let's look at conjugation. To conjugate means to come together. If you were unfortunate enough to be in prison for a very long period of time, you might look forward to a conjugal visit. Now, when we look at bacteria, we see that it has its main chromosome, this big circular chromosome that has all the genes necessary for its growth and reproduction. But sometimes bacteria will have extra smaller pieces of circular DNA called plasmids. And these plasmids will carry extra non-essential genes. But not all bacteria have a plasmid. Sometimes a bacteria cell with a plasmid will transfer a copy of that plasmid to another bacteria. So here we have two bacteria, one with the plasmid and one without. You can see the one that has the plasmid has some characteristics, in this case these proteins on the surface or something, that are being conferred by this plasmid. And this one does not have the plasmid. It's almost like we have two different versions of the bacteria. We won't go as far as saying different genders, but they are different. So when they come together, they'll form what's called a connection, what's called a conjugation tube. And through that tube, we're going to exchange, basically like downloading a file, we're going to exchange part of this information, part of this plasmid. So half the DNA plasmid ladder is transferred across. As it comes across, on the new side, we're going to build the other side of this double ladder. So we can watch this process happening. And as we transfer this across, at some point this conjugation tube is going to break and kind of end our download transfer. And it's not uncommon for that download to finish, or for that conjugation tube to break before the download has finished, before we finish transferring the entire plasmid, so that we get a little bit different variation, and so that this bacteria might get a slightly different version of the plasmid and therefore express a slightly different version of the trait. So notice that bacterial conjugation did not create any new bacterial cells for us. It's not reproduction, but it does introduce genetic variation into our bacterial population. So bacterial conjugation, the sharing of a plasmid from one bacterium to another, creating no new bacteria, but introducing some genetic diversity into our population. Now let's look at bacterial transformation. To transform means to change. It turns out that bacteria have the ability to pick up DNA from their environment. So here are two plasmids sitting out here in the environment. They may be remnants of some bacteria that have died, but regardless, they're out here and free and available. And bacteria have the ability to move in and pick up and incorporate these plasmids into their cells. And then they have the ability to express the genes on them. Therefore, they are changed. They have transformed. Not only that, when these cells go on to reproduce through binary fission, they'll copy these plasmids and their offspring will also have these extra genes. This process of transformation is actually very important in our understanding of genetics and in terms of determining what DNA was. One very famous experiment by Frederick Griffith showed that the different strains of pneumonia that he was experimenting with were transformed by genetic material. We'll discuss that experiment later this year. So again, bacterial transformation, picking up plasmid or any DNA from the environment, it is not creating any new bacteria for us. It's not reproduction, but it is introducing genetic diversity into our bacterial population. Now let's move on to bacterial transduction. Now if you recall, let's look at what the word transduction means. Trans means across, and duction or to conduct means to move across. Bacterial transduction is the movement of bacterial DNA from one bacterium to another via a viral vector. So we need to remind ourselves of the lysogenic viral replication cycle that we learned about not too long ago. So here's our bacteria cell, and we need a virus to infect it. So here's our bacteriophage, our virus that infects bacteria. Our viral DNA will infect the host cell. We can watch that process happening here. And the viral DNA is going to insert itself into the host DNA for a latent stage, so let's watch that happen. And we know that this viral DNA, now that it's hidden into the host cell's DNA, can remain hidden here for a long period of time and may even go through some passive viral replication. But at some point, this viral DNA is going to excise itself from the host DNA. And when it does, sometimes it can pick up some of the bacterial DNA with it. We can see here the red is the viral DNA and the white is the bacterial DNA. Well, the next step in our process is to construct the viral components, so let's build those. And then finally, we actually assemble our viruses. And watch what happens here, because something very interesting could happen. We see that in this virus and then this virus, or these protein coats of the virus, we have incorporated some bacterial DNA. Some bacterial DNA has been packaged into these viral protein coats. We can see them, let's see, right there. Now eventually these cells are going to burst or lyse to release these viruses to go on and infect other cells. Now notice that some of these viruses, like these two, are carrying bacterial DNA that they can transduct or transfer over to other bacterium. So let's watch that process. Let's bring in two bacteria, and these two viruses, these two bacteriophages, are going to infect these bacteria cells, and they're going to insert this DNA into the host cell's DNA. So again, transduction is not bacterial reproduction, but it is a source of genetic variation in bacteria. We've moved these white genes from one bacteria into the genome of this bacteria, which may have a whole different set of genes. So it is an important source of genetic variation for us. So we can summarize transduction as, or describe it as, when a virus relocates bacterial DNA from one bacterium to another via the viral replication cycles, introducing some genetic diversity into the population. So there we have it. These are the four processes you need to know about bacterial reproduction and exchanges of genetic material to explain how viral populations can become genetically diverse. So go back over each of these, draw lots of pictures, kind of be able to explain each step along the way, and make sure you can differentiate between these four. If you have any questions, leave them in the comments below the video, and I hope you learned something.